Welcome back to the course on Polymeric Biomaterials. We are now looking at the topic of biomedical polymers, and in today's lecture we will be continuing our discussion on synthetic biomedical polymers that we began in lecture 17. In the previous lecture, lecture 17, what we did was to set the stage by developing what we called structure, property, function relations. Of course, all of this is affected by processing. For polymeric biomaterials in general, and then what we did was to use this paradigm to see how in specific clinical applications specific types of polymers are used. So, we asked what is the demand in the clinical application, and then we said why is a particular polymer chosen for that application based on the structure, property, function paradigm. Now, in this lecture what we will be doing is turn our attention to some very specific biomedical polymers that are clinically important. What we will do here is focus on these polymers and again look at how structure connects to properties to function and how all of that can be affected by processing. And then see how all of that in turn determines how these polymers can be used in specific applications. So, we will be looking at specific polymers, namely polytetrafluoroethylene, polyethylene terephthalate, so this is PTFE, this is PET, polymethyl methacrylate or PMMA, polyethylene glycol which is PEG. Polyanhydrides are a class or a family of polymers which we will look at, and then towards the end we will focus on degradable polyesters. Some very popular examples that we have seen so far in the course include polycaprolactone, polylactide, polyglycolide, copolymer PLGA, and then we will end the lecture by looking at one important study where we look at some of these polymers and see how they can be employed for controlled drug release. So, we will first begin with polytetrafluoroethylene or PTFE. Now, PTFE was initially discovered by accident in some sense at DuPont by Roy Plunkett, who was a chemist who was working at DuPont, and he was working with tetrafluoroethylene for some other application, and the story goes that one day he found that in a cylinder of TFE he found this white powdery smooth substance which later on came to become PTFE. So, the TFE had actually polymerized within the tank, and that was an accidental serendipitous discovery which was very important. This was done in the 1930s, and 1930s was also important because another very important industrially important polymer which was also then subsequently repurposed for clinical applications was prepared and fabricated, and that was if you recall from our earlier discussions was nylon, and the scientist there was Carothers, who incidentally was also working at DuPont. So, that period 30s, 40s and 50s were a very important period for the development of many of the polymers that we still continue to use today. So, with that quick very brief history of PTFE, we now move on to looking at structure property relationships in this polymer. So, the first thing we will look at is how microstructure affects properties, and when I say microstructure, we look at it at two levels. The first level is the individual chain level type of structure, and then we will look at also how those can in turn help affect the microstructure at the bulk. So, we first note that this material is very unique because of the very high strength of CF bonds, which in turn confers exceptional thermal and chemical stabilities for PTFE. It also so happens that the very small size of F and the repulsion of adjacent fluorine atoms results in a twisted helical chain as shown here. Now note that you will not find this kind of twisted helical chain for polyethylene, where you only have hydrogens instead of the fluorines. So this is unique to PTFE because of the CF and the presence of fluorines along the chain. Now what also is a consequence of that is the fluorine shell, if you will, sort of encases the inner carbon backbone. So you see the carbon is barely seen. The carbon backbone in the chain is sort of hidden away and sort of protected by the fluorine atoms that go along. So because of this encasing, the carbon bonds, the primary covalent bonds along the backbone are protected from reactions. So this is one of the reasons why PTFE is extremely stable. Of course, none of this is obvious when you simply draw the structure this way, which is one way to represent the structure, no doubt, but some of these are much better visualized when you draw the structure this way. So at a higher level, so now we are moving up in the hierarchy, and now would be a good time to recall some of the ideas we have seen in earlier lectures about hierarchical structural organization and polymers, where the organization at different length scales sort of build to result in the bulk properties. So one consequence of having this helical chains is they pack as solid crystallites, resulting in long, parallel and stiff molecular rod-like structures. And because of the very tight packing, that leads to very high bulk crystallinity. Right. So you see how beautifully the properties are connected, and you can go from step 1 to step 2 to step 3 and sort of logically see the progression of how the structure evolves over multiple length scales. And as a consequence of very high bulk crystallinity, it is going to scatter visible wavelengths, which means you are going to typically see opaque white kind of material. Now if you remember, I mentioned that Roy Plunkett, when he first discovered PTFE, he found that it was very smooth, and one reason for that is because of very low chain interaction energies, resulting in very low coefficient of friction. So the coefficient of friction being low means that the surface is going to be extremely smooth. The material is also non-polar with low interfacial energy and hydrophobic, which also explains why PTFE is a very popular polymer for your non-stick ware. Non-stick vessels that are supposed to repel water are largely coated with this material. But for biomedical and clinical applications, they are more used for catheters because of some of these properties, right from inserting the catheter to its function, its interactions in the body, etc. This is a very suitable material, but note that just because it is used in catheters does not make it suitable for other kinds of applications. For example, it is not a very good polymer to be used for bearings or joints because of two reasons. One is it is susceptible to creep. It is a property that we will look at in more detail in subsequent lectures, and it has very poor wear resistance, and this is something we have seen when we looked at the Vitek implant case study in lecture 5, where we saw that the inappropriate use of PTFE for implants that were intended to sort of augment or replace the temporomandibular joint led to wear debris with time, and that is because PTFE has very poor wear resistance. But from a material perspective or a processing perspective, there are other limitations of PTFE. One is a very high molar mass and rigid helical conformation, resulting in very high melt viscosities, which makes processability very hard. Now this is something that we have seen again when we discussed molar mass, when we saw that melt viscosities can become very high with increasing molar mass, but with increasing molar mass your mechanical properties also go up. So there is always a sweet spot that you find in between where you can balance out the processability with reasonable viscosities without compromising mechanical properties significantly. Another difficulty with processing is the very high melting temperature, which is not too far away from the degradation temperature. So, if it is not carefully controlled, you can quickly end up degrading the material. Now, commercial versions of PTFE sort of overcome these limitations, and we will look at two important examples, one from DuPont, one from Gore. And we will see how processing the material differently or changing the structure a little bit can result in slightly different properties, making it more processable, which also helps in clinical applications. So, the first example we will look at is this material called Teflon FEP, which is developed by DuPont. Essentially, FEP is fluorinated ethylene propylene, and it is produced as Teflon FEP primarily to reduce PTFE's very high crystallinity and melt viscosity. So, you have to be able to connect the very tight packing of chains to very high crystallinity to very high melting temperature and very high melt viscosity. So, all of those are connected. And the way this is done, or the way this melt viscosity can be reduced, is through the introduction of perfluoromethyl groups, which create defects as shown here. So, the introduction of these groups every now and then introduces defects in the crystallites, meaning the chains are not as tightly packed, which now ensures that you can reduce the melting point quite a bit, and it also reduces chain slip and creep, which are again problems we saw with traditional PTFE. So, in one sentence, FEP has similar properties as PTFE. So, while all other properties of PTFE are reasonably not compromised, it also helps with processability. So, that is one way to overcome the limitations of PTFE that we saw earlier. And of course, shown here is the structure of Teflon FEP, where you see the traditional group that is there, plus the introduction of the perfluoromethyl groups, which at occasional points, which will disrupt the ability of chains to pack very tightly, which results in lowered melting point, better processability, lowered viscosities, etc. Another important commercial version of PTFE is this expanded polytetrafluoroethylene developed by Gore. Now, there is also an interesting story to how this was developed. It so happens that Bob Gore, who was the son of William Gore, he was trying to see if he can improve the properties of PTFE by applying very high strain rates at high temperatures, and he saw that resulted in a microporous structure, which was later called expanded Teflon. So, ePTFE, because of its microporous structure, is more flexible than PTFE and also allows permeability, but without compromising strength. So, this microporous structure also allows tissue ingrowth and anchoring when used as vascular access grafts. So, essentially these AV grafts, as they are called, are essentially shunts that are placed for drawing out blood and purifying it outside and sending it back to the patient for people who have lost their kidney function. So, this is what is called dialysis, and in such patients these AV grafts that are implanted are typically these ePTFE. And this is also an excellent material because these grafts have to be accessed every other day, or once a week or twice a week, depending on the need for dialysis, and they will so they have to be able to withstand multiple pricks, etc., with a needle, and they perform very well on all those fronts. Some of these grafts are also pre-clotted to prevent leakage after placement, which is you take the patient blood. This is sort of an older technique where you take the patient blood and pre-clot the graft to make sure that you can prevent leakage after implantation, which limits thrombosis. But many of these grafts are also what you call heparinized. Now, heparin is a molecule that is a natural anticoagulant. And heparin's structure and its properties are things that we will discuss in great detail in a subsequent lecture under natural polymers, but we will simply note here that heparinization is a very common technique to ensure that surfaces do not clot. In fact, sometimes when blood work is done and blood is drawn from your veins and they take them in vials, and if they should not clot, sometimes the surfaces of those vials inside are either coated with heparin or EDTA. This prevents or slows down clotting. Anyway, so heparinization is one technique to make surfaces non-thrombogenic, which means they are not going to clot, which is something that you would desire in an application like this. Now, that is PTFE. We will next move on to polyethylene terephthalate. Now, this is another polymer we have seen in an earlier lecture in the context of polymer synthesis, which is lecture 11. And it is a copolymer. We have both the ethylene and the terephthalate groups. So, ethylene glycol and terephthalic acid, those are the monomers that are used to form this copolymer. And note that from a polymer family perspective, it is a polyester. So, the ester bond is present. It is a semi-crystalline polymer, which means it is going to exhibit both a Tg and a Tm. So, you will have a peak melting temperature of around 250 plus and a glass transition temperature around 70 degrees. And what is important here to note is typically when it is used in applications, it is extruded and drawn, which makes it very strong and flexible, being stabilized by the high crystallinity, chain rigidity and orientation. And although it is a polyester, and the word ester should immediately bring to your mind the fact that these kinds of polymers can potentially undergo ester hydrolysis and degrade. But because it is very highly crystalline, the degradation rates are very low compared to other polyesters such as polylactide or polyglycolide or PLGA. And a commercial version that you might be familiar with for PET is called Dacron. Now, like PTFE, Dacron is also used quite a bit for large diameter vascular prosthesis and arterial grafts or shunt, primarily because it has very high elastic modulus and tensile strength. But one limitation with PET is its low compliance. Now, what that means is the elastic recoil of PET is quite different from those experienced by native arteries. So, this mismatch in compliance, what that leads to is it can lead to disruptions in the blood flow, which can eventually lead to graft failure. So, very often woven or knitted forms are used, which overcomes some of these limitations. So, they also, apart from providing durability, the porosity like we saw for the expanded PTFE can also support tissue ingrowth and endothelization. Now, endothelialization is a process where a surface can be covered by endothelial cells, which are naturally present in all your blood vessels. They line the inner wall of the blood vessel and they act as natural anti-thrombogenic surfaces. So, if a surface can get endothelialized, then that is going to ensure that the surface becomes non-thrombogenic. They are also used as non-resorbable sutures, and note that they are not going to degrade very easily. That has to be connected to the fact, to the structure, to a bit of hydrophobicity and to the crystallinity. And the sutures are typically extruded multifilament fibers and braided. The structure and the properties ensure that they are reasonably biostable for many years. But there are concerns about degradation. After all, it is a polyester. So, if there are defects during processing, etc., there could be concerns about degradation in long term. And shown here is knitted crimped PET vascular prosthesis with type 1 collagen impregnation. So, that is PET. So, we have looked at PTFE and then now we have looked at PET, which is a polyester copolymer. We will next move on to a non-crystalline amorphous polymer. So, what you see, we are trying to look at different classes, and between the polymers we are trying to cover several of the concepts we have learnt earlier in terms of structure, and to see how those properties for those polymers directly impact their biomedical applications. Polymethyl methacrylate or PMMA, as it is more popularly known, or plexiglass if you will, which is one version of it, is an atactic polymer, which means largely it is going to be amorphous. So, the fact that it is atactic, it is going to be connected to its amorphous nature, and also because it is amorphous, it is going to be transparent, and that has to do with how the optical properties are dictated. It is strong and lightweight, and it is typically used, at least the application we are looking at here is that of bone cement for hip implants. And the story for how this evolved is also interesting. So, Charnley had approached Dennis Smith, and this is a story we have seen in the previous lectures. I am just quickly recapitulating this to look at the use of acrylate based polymers for cementing materials, and that eventually led to PMMA or polymethyl methacrylate being used for bone cement for hip implants. So, essentially this is what you would call grouting, if you will, right. So, it creates a tight space and holds the implant against the bone. It is able to fill all the micro cracks, etc. Note that it is intrinsically not an adhesive polymer. It forms an interlock between irregular bone and the prosthesis. So, any cracks, micro cracks, etc. are sort of bonded between the bone and the implant through PMMA cement. And shown here is an X-ray of a cemented total knee arthroplasty, where the arrows indicate the bone cement, right. Now, of course, this part is the knee implant itself, which again we have seen in great detail in previous lectures. Now, from a structure property processing perspective, one can ask, how is this deployed in the clinic? So, typically it is supplied as a two-part kit, like silicones that we have seen earlier, where it is supplied as a two-part kit, and PDMS, for it to be cross-linked and formed, the components have to be mixed on site and cured for it to cross-link. Likewise, PMMA is also supplied as a two-part kit, and there are multiple components to each of those parts. So, there is a powder component and a liquid component, and within the powder component there are some 4 or 5 subcomponents, and within the liquid there are some 3 or 4 subcomponents, and these are provided separately. So, they are usually mixed on site, and then the process of cross-link or polymerization starts. Also, one thing you notice here is these different components that are there serve different functions. So, it is important to note how multiple aspects are integrated into one system to be able to give it the multifunctionality that you want. So, the polymethyl methacrylate itself forms the bulk, that powder itself forms the bulk of the bone cement. The benzoyl peroxide acts as the initiator, initiates the process, and barium sulfate is typically added as a radio opacifier, so that you can see this in X-rays. To prevent bacterial infections during surgery and post surgery, antibiotics such as gentamicin can be integrated. Now, in the liquid part, what you have is the monomer methyl methacrylate, which in the presence of the accelerator, this NN dimethyl para-toluidine, which also initiates the free radicals and accelerates the reaction, reacts to form and set as bone cement. Now, would be a good time to recall free radical polymerization concepts that we have seen in lecture 11 earlier. Now, to ensure that this does not prematurely cross-link while still in the liquid state in the bottle, a stabilizer such as hydroquinone is added. So, shown here is a schematic of that process itself, where the liquid components are mixed with the powder components and additives could be added. So, one possible additive that could be added could be some kind of a ceramic, calcium phosphate for example. Typically, it is calcium phosphate based ceramics. And the reason to do that is you can now make the PMMA bioactive. On its own, PMMA will only serve a mechanical purpose and a physical purpose. It will not be bioactive on its own, but if you want to make the system bioactive in the sense that it can actively interact with the biological host, then you can add additives like calcium based, magnesium based oxides, phosphates, etc., which can help the bone cement to become bioactive. So, this is a good example of how a material that is not intrinsically bioactive can be made bioactive. Can you recall another example that we have seen earlier of where a material that is intrinsically not bioactive has been made bioactive? If you are able to recollect, you will recall that titanium implants, which also serve primarily a mechanical function, can be surface coated or spray coated with ceramics to make them what we would call osteoconductive. So, here what happens is the liquid components with the powder components and the additives are mixed in situ and the free radical polymerization proceeds on site and ensures that the bone cement sets. So, that is how PMMA works. So, you see it is a nice stepwise progression from understanding why you have these methacrylate groups. And again, wherever we have seen that they can undergo free radical polymerization in the presence of free radicals, to how the properties of the polymer eventually guide how they are used in this particular application. We will next move on to another very important polymer, namely polyethylene glycol or PEG. From a structure property perspective, this is simply a linear water soluble hydrophilic polymer, and its structure is simply given by this CH2 CH2O. Of course, it is a polyether, the structure should tell you that, right. So, wherever possible, please note the family of polymer that we are talking about as well. It is extremely hydrophilic and it can extensively hydrogen bond in the presence of water, and that in turn has consequences for what types of applications PEG can be used. PEG is also interesting because it has molar mass dependent properties, which means you can have PEG go from very low molecular weights where they exist as liquids below about 1000 gram per mole, where it would barely classify even as a polymer, to waxy solids and flakes and powders at higher molecular weights. So, depending on the molar mass, you can have different physical properties going from liquid like properties all the way up to flakes, powders at much higher values, not unlike silicones, where the properties depend on molar mass and cross linking in that context. And this is just a subtlety, but it is an important point to note. You will also find PEO being used in the literature. When you read papers, look up textbooks, you will find polyethylene oxide being used, sometimes interchangeably with polyethylene glycol. Now, structure wise they are exactly the same, but PEG typically over a certain molecular weight goes by the name polyethylene oxide. So, if it is below a certain molecular weight or thereabouts, it is called PEG. If it is above a certain molecular weight, it typically goes by PEO or polyethylene oxide. That is an important subtle point to note. So, when you see people using it interchangeably, then you may want to stop and ask what is the molecular weight that they are talking about. Now, These specific sets of properties we have seen, particularly its hydrophilicity and its ability to extensively hydrogen bond, dictates its applications. One very popular clinical application is oral ingestion, where PEG 3350, which is simply the molar mass, has been FDA approved for use as laxatives in adults. This is also used in colonoscopy preparation. This is because of its high water binding capacity. It does not get absorbed in the intestines much, very limited adverse effects in adults at least, and low toxicity. So, all of these factors ensure that PEG 3350 can be used as an adult laxative, which is FDA approved. Two things to note here are, one is the phrase FDA approved, which is different from FDA cleared. The bar for what is FDA cleared is much lower compared to the bar for what is FDA approved. The two are very different, and we have seen this partially in the context of the Vitek implant case study that we saw much earlier in this course. If you are not familiar, I would strongly encourage you to look up the difference between what is FDA approved, what is FDA cleared. That is one point to note. The second important point is, FDA approval is often sought or always sought in the context of a specific material or a device used in a specific application. So, it is incorrect to say PEG 3350 is FDA approved. The more correct way to say it is, FDA approval has been received for PEG 3350 for use as adult laxatives with those specifications. So, that is what FDA approved actually means. Instead of saying that a material intrinsically or per se is FDA approved, what is more correct to say is it is FDA approved in the context of a specific application. That should also explain why, although a certain material may be FDA approved in one particular application, it needs to go through the process again if it is used in a different application in a different part of the body, because the interactions are very different. That is primarily the reason. And FDA or agencies such as the FDA are primarily focused on safety and efficacy. Both of these are important, and when they do clinical trials, these both will form the basis for whether something gets approved or not, compared to existing gold standards. Now, one very important application of PEG is its use as surface coatings and in anti-fouling applications. Now, the way PEG is used in such applications for surface coatings is as grafted chains on surfaces. Now, if you recall our earlier lecture, lecture 10, you will remember that we have looked at brushes as being one type of polymer architecture. So, that would be a good place to go back and refer if you want some more details on what we are talking about here. Nevertheless, once they are grafted on surfaces, both chain length and surface density become important. And here chain conformation plays a very important role, and it so happens that chain conformation is a function of grafting density. Again, chain conformation is something we have looked at in lecture 9 in very great detail. Now, how does conformation depend on grafting density, and why does it matter? That is the question we will ask here. Shown here are cartoons of two different grafting densities where the chains take up different conformations, where D is simply the distance between two grafted chains. If the distance between two grafted chains here is above what you would call the Flory radius here. Now, this is not something we have looked at earlier, RF, but what we have looked at is RG, which is one way to think about size of a macromolecule in solution. So, the Flory radius is simply one way to think about size of a polymer chain in a good solvent based on Flory's mean field approximation theory. But for the purposes of this discussion, one can imagine that it is not very different from RG, although they are not the same. In this limited context, you can use Rg as one way to think about what we are talking about here. So, if the distance is well above the Flory radius, then the chains exist as random coils, giving this mushroom like structure. That is why it is called a mushroom. Now, if the chains are placed very close to each other, where the grafting density is very high, then because of extruded volume effects, these chains will stick out like a brush, as shown here, where this distance between them is lower than the Flory radius, resulting in a dense brush which happens at high surface coverage. So, you see, intrinsically we are not saying anything about how PEG will behave on a surface, it depends on how they are grafted, right. So, two different grafting densities, these are sort of the two extremes, where in one case you get the random coil conformation, where again now would be a good time to recall the scaling laws for ideal and good solvents that we have seen earlier. Ideal here simply refers to theta, the theta condition, and good solvents we have seen scaling laws for these in lecture 9. And in the other case, where the grafting density is very high, essentially where the distance between the chains is lower than the Flory radius, then you end up getting a brush like conformation. And in this case, the brush height L at surface in a good solvent for high chain density is given by this expression here, where L is approximately equal to n times A times sigma to the 1/3, where A is the monomer size, n is the monomers per chain, and sigma is the number of chains per area. And this is largely due to the Alexander-Dejen theory, where Dejen develops in a paper in 1980 in macromolecules this theory. Now, one way to think about this equation is to ask, does it physically make sense when you look at this equation? So, when you come across new equations, one thing I would encourage you to stop and ask is, can I make physical sense of it? So, the length of the height of the brush height intuitively should depend upon how long the chains are, which is given by this, and how closely placed they are, which is given by this, which affects the conformation. So, you see, this is one intuitive way to understand how some of these equations can be physically visualized. So, this is a good example of how PEG is used for surface coatings. Now, we still have not answered the question of how this results in repelling proteins and antifouling properties, which is what we will look at next. So, PEG's role in antifouling primarily comes from its immobilization on surfaces, as shown on the previous slide, which reduces protein adsorption. Now, this is a fairly complex set of mechanisms with both enthalpic and entropic components, which we will look at in more detail in a later lecture when we do surface properties of polymeric biomaterials, where we will look at mechanisms of protein adsorption. But suffice here to say that it primarily comes from the hydrophilicity of PEG, where the bound water ensures that any incoming protein has to displace the water. So, that is not favorable. There are of course steric hindrance factors also, and it is a combination of factors that ensures that PEG grafted surfaces at high densities will prevent protein adsorption. And if proteins do not adsorb onto surfaces, or they adsorb in very negligible amounts on surfaces such as this, where incoming proteins do not adsorb as much, then cells cannot recognize the surface or cannot attach to the surface, because cell attachment to surfaces happens through proteins. So, and if proteins do not adsorb, the surface becomes non-fouling. But one caveat to note is many of these studies, especially in the earlier days, were done in vitro by exposing these functionalized surfaces to serum or mixed controlled protein solutions, etc., where you got really good results. But that reduced adhesion or adsorption did not always translate to lower material thrombogenicity. So, any promising results in controlled in vitro studies did not always translate to in vivo results. Now, this is an important point to keep in mind. It's a good example where we see that not everything that works very well in vitro will translate in vivo, because in vivo in the body the mechanisms are far more complex. The way the body functions, the way the body recognizes the material, interacts with the material, etc., everything will change as compared to how cells interact with surfaces or proteins interact with surfaces in very controlled microenvironments in vitro. Now, that does not mean in vitro studies are useless. They are very, very useful for two reasons. One is they provide directions they are good indicators of whether something will or might perform well in vivo. The more important reason for in vitro studies is also for us to understand mechanisms. So, for people who are interested in probing very specific mechanisms under controlled conditions, where the goal is not really to apply something but to understand the system, these controlled in vitro environments are very, very helpful. But limitations are that PEG may not be very stable long term in vivo. So, any grafting that is done may eventually go away with time, and it can also be metabolized or undergo oxidative degradation, which can also be problematic when you want to use it for long term antifouling applications. Another way to think about surface grafting from a chemistry and a commercial perspective, you can now look at surface functionalization with PEG through covalent attachment. Now, the way this is grafted, that's science in itself, and there are extensive studies that are done. Also note that here we are looking at flat surfaces. You can also have microparticles or nanoparticles that are grafted with PEG chain. These are what are called polymer grafted particles, and they have a variety of applications in biomedical engineering. So, their curvature effects will also play a role, which we are not accounting for here in flat surfaces. At any rate, the way the PEG is attached to the surface is through one way to do that is through covalent bonding by derivatization of its hydroxyl N groups with an active coupling agent. Some commercial or popular examples of that include amino PEG, NHS PEG, etc. Although we are not going to get into the chemistry of these kinds of surface functionalization, I would encourage you to look up the ADC NHS chemistry, which is a very popular way to functionalize surfaces with different kinds of molecules and change the surface property. That is essentially the role, right. The role that they play is to change the surface chemistry and in turn change or alter the surface properties, and surface properties are equally important, if not more important than bulk properties, especially in biomedical and clinical applications. So, one example of that we are looking at is a coating, a commercial coating for cardiopulmonary bypass circuits. So, these cardiopulmonary bypass circuits are essentially systems that where heart surgery is being done, they temporarily take over the role of the heart and lung, where they will help flow the blood through and oxygenate the blood as well, where the heart is temporarily quote unquote stopped to aid surgery. In such cases, these kinds of machines will take over, and because the blood is flowing, you do not want the system to be thrombogenic, you do not want clotting to occur. So, typically what happens is the biomaterial surface is typically functionalized first with a hydrophilic priming layer, and then you have these PEG chains that are covalently bound along with negatively charged sulfonate groups, and then of course to aid or to confer more antithrombogenicity function, they are also covalently attached with heparin. So, you have PEG, you have heparin and you have sulfonate groups which are negatively charged. So, this hydrophilic negatively charged surfaces mimics the vascular endothelium, which is the natural inner lining of blood vessels which prevents thrombosis. So, this kind of a functionalization minimizes protein adsorption and reduces thrombus formation, which is a very critical function that has to be taken care of during the use of this cardiopulmonary bypass. So, you see how the properties like hydrophilicity and the negatively charged groups coming from sulfonate can, and the heparin as well, together play a role in determining how these materials perform in specific biomedical applications. Now, having looked at some of the more classical polymers, we will now move on to other types of polymers that started being developed around the 80s and 90s, or got very popular around that period for biomedical and clinical applications. We will start with polyanhydrides and then move on to degradable polyesters. Now, polyanhydride is the structure of which is shown here. The anhydride linkage, one very important property of it is that it has very poor hydrolytic stability, and it can sort of cleave in days to weeks. And typically polyanhydrides are synthesized from hydrophobic aromatic or aliphatic diacids. And note that polyanhydrides, the name itself indicates a family of polymers, and depending on what the individual monomers are, etc., you can get a series of different types of polyanhydrides with slightly different properties. These undergo what you would call surface erosion and resulting in diacid monomers that can be metabolized by the body, and the degradation rates can be controlled. So, shown here is one example of a polyanhydride, which is a very popular copolymer, poly-CPP co-assay, where CPP simply refers to the bis-carboxyphenoxypropane and assay refers to sebacic acid. Now, this polymer is interesting for a bunch of reasons. First thing you note, we have already said that degradation can be controlled very carefully through the choice of monomers, and hydrophobic monomers will clearly have a slower rate of erosion compared to hydrophilic monomers. So, just as a comparison, if you see pure CPP, right, will take 3 years to degrade, whereas the inclusion of the assay with 80% assay now brings down the degradation time frame to the order of days to weeks. So, you see pure poly CPP takes much longer to degrade, while the copolymer with the sebacic acid brings down the degradation rate to a few days to a week or 2 weeks. And now that is a very interesting way to tune or tailor degradability. Why is this important? It is important because these can be used in controlled release applications. So, short term drug release where you can achieve near zero order drug release from thin discs made out of this polymer can help in many applications. One application we are going to see next, but before we do that, I would encourage you to look up the history of polymer development and application for controlled release in the period of 80s and 90s, largely due to the work of Professor Robert Langer and co-workers, developed a series of polymers and tested out other polymers for controlled release applications, polyanhydrides being one of them, polyorthoesters, polyesters like PLGA, polylactic acid and so on. So, it is a very interesting period in biomaterial development where many of these polymers were developed for controlled release applications, both surface eroding and bulk degrading. And here we are looking at surface eroding polymers. So, the tunable degradability like we saw on the earlier slide ensures that you can get very specific types of release profiles of drugs which are embedded within these polymers. And one commercial clinical deployment of that happens to be these gliadel wafers. So, this is FDA approved for malignant glioma which is implanted in brains of patients. Now, essentially it is this copolymer that we saw on the previous slide, which CPP and sebacic acid 2080 loaded with this particular drug, carmistin. Now, the fact that this drug is loaded into the polymeric matrix ensures that the drug itself is protected from hydrolytic degradation before it is released. We note here that the half-life of this drug is only 52 minutes in the body on its own, but once it is loaded into the polymeric matrix, the polymer ensures, or the matrix ensures, that the drug is protected from degradation until its release. So, that is a very important idea to keep in mind. Now, the other reason why this drug is put into this material before it is implanted is these materials undergo what we call surface erosion. Now, we are going to look at polymer degradation in a separate lecture, where we will look at degradation mechanisms in more detail, but for now we will simply note that this is surface erosion as opposed to bulk degradation that some of the polyesters which undergo ester hydrolysis will experience. So, the surface erosion of this polyanhydride ensures that the release is sustained without a large initial burst, because if you want to release a drug, your limits are, this is the toxicity limit, this is the minimum effective concentration, and this is what you would call a therapeutic window. So, any matrix that releases a drug has to ensure that the drug concentrations are in the released form are retained largely within the therapeutic window. You do not want to go beyond this, because that is going to lead to toxic effects. Anything that is below this is not going to be effective. So, you want to ensure that when you design drug delivery vehicles, this aspect is kept in mind. And it so happens that this avoids the initial burst, which avoids any potential toxic effects, and also provides sustained release to have long-term therapeutic effects. And so, the slow release mitigates overdose and prolongs the action of the drug. So, once the gliadel wafers are implanted once, which are typically implanted following brain surgery for malignant glioma, before the surgeons close up the head, these wafers are inserted, and the slow release of this drug kills the cancer cells over a few weeks. And it is fully resorbed by the body, because the degradation products are simply diacids. It can be fully resorbed by the body, which avoids another second surgery for removal of the wafers. And if you want to see what that looks like, here is a picture of that, where you see these drug loaded wafers, polyanhydride wafers, which is gliadel, being implanted as thin discs in the brain following surgery and removal of tumor. Now, one aspect that I want to highlight here is that here we want to look at how processing determines performance. Now, the finished form of a product is also very important. When we talk about processing, the finished form is also very important, because in a application like drug release, the surface area to volume ratio becomes very important, both for degradation kinetics and for the release of the drug. Typically, larger surface area leads to higher exposure to water, leading to faster degradation. So, shapes with very large surface area to volume ratio typically include fibers, nanoparticles, thin slabs and porous scaffolds. So, instead of placing one large wafer which could take a very long time to release the drug and keep it in the effective therapeutic window, typically about up to 8 small and thin wafers are implanted to ensure that you get enough drug to be released for it to be effective in that part of the body, right. So, that is what is said here. after tumor removal, up to 8 wafers are deposited along the wall of the resection cavity, and shown here is a picture of that. Next, we will move on to bulk degrading polyesters. Now, degradation again here means irreversible change in the structure of the material or fragmentation, which is typically manifested as a significant change in properties of the material with time. So, as the material degrades, properties like molar mass will change, its mechanical properties will change, lots of other things will change. And again, we will look at those mechanisms in more detail in a subsequent lecture, but for now we will note that we are here talking about bulk degrading polyesters. And in the biomedical context, it simply means a reduction in molar mass under physiological conditions. So, the backbone, you say reduction in molar mass, just this, the backbone itself degrading, or the backbone itself undergoing hydrolysis, because they are esters. Now, the advantage of this is that it is for a tissue engineering application, for example, with degradation, these materials can be replaced with neo tissue, and a second retrieval surgery is not required, because if you can control the rate of degradation of the material to match with the tissue regeneration rate, then by the time the tissue is fully regenerated, the material is going to completely degrade. But of course, one drawback with this is the release of degradation products, which again we have seen earlier have to be carefully chosen such that the degradation products do not cause any adverse reactions. And some popular examples of these bulk degrading polyesters include polylactic acid, polyglycolic acid, PLGA copolymers, polycaprolactone. Many of these we have seen in different contexts in earlier lectures. A lot of these polymers are synthesized using ring opening polymerization reactions in the presence of metal catalysts. So, you see here polycaprolactone we are showing here, polyglycolic acid we are showing here, and polylactic acid we are showing here. So, these rings are opened up during polymerization, leading to polyesters. All of them will have the ester linkage. So, these polymers are very important and quite widely used for biomedical applications. We will look at the properties of some of these polymers and then finally look at one application of them in controlled drug release. So, firstly note that you get a wide spectrum of properties. When I say properties, I include the microstructure, the glass transition, the melting temperature, and the degradation. Many of these properties we have dealt with in much detail in previous lectures. Now, degradation time is shown here in months, and that can depend on a lot of factors. And in turn, a polymer with slow degradation rates or fast degradation rates are preferred depending on the type of application. In some cases, you want the polymer to degrade very quickly. In some cases, you want the polymer to remain in place for a very long period of time. And some important factors that play a role in degradation include hydrophobicity, the N group. We saw in PLGA, for example, if you have a ester terminated PLGA versus an acid terminated PLGA, you will have differences in degradation rates. Molar mass, typically much higher molar masses are preferred when you want the polymer to be more stable or the material to be more stable and not hydrolytically degrade very quickly as compared to lower molar mass. Crystallinity, again we have seen that if you have highly crystalline materials, they will degrade much slower compared to amorphous materials. Likewise, tacticity, porosity and surface area are very important factors for determining degradation. Now, I have simply listed out some 5-6 polymers here, and you see even within these 5-6 the extremely wide variety of properties you can achieve, going from semi-crystalline polymers to completely amorphous polymers, and you get a range of glass transition temperatures and a range of melting temperatures and a range of degradation time periods. And note that properties like glass transition to a larger extent and melting temperature to a lesser extent are given as ranges because they depend on many other factors. Now, based on these properties, typically PCl is used in sustained drug release. It degrades very slowly. So, when you want to have very long release time periods, polycaprolactone is used, and one commercial version that showed a lot of promise in clinical studies was this contraceptive Capronor, which released this levonorgestrel over 12 to 18 months, and it was a subdermal contraceptive. It was never really commercialized significantly because of competition from other players, but also retrieval was a bit difficult and it had very long tails for release. But anyway, what we are trying to say here is that the sustained drug release has actually been employed for developing a device that can release a certain drug at a certain rate over a very long time period, 12 to 18 months. PGA, for example, is used in orthopedic fixation screws and sutures, and with time they will resorb, ensuring that unlike metal pins or metal screws, you do not have to go back and sort of retrieve them or correct them, they will resorb with time. Of course, PLA, PLGA are used in drug delivery. We have also seen the use of some of these polymers in face fillers, where for dermal fillers, they will initiate a small localized immune reaction which will lead to deposition of collagen, which is what is desired in those kinds of filler applications. And for drug delivery, we are looking at another example of a commercial implant which releases this corticosteroid called dexamethasone for up to 6 months or longer in the eye. This is used to control chronic inflammation in patients who are suffering from a condition known as diabetic retinopathy. So, these are PLGA based rod-like materials loaded with dexamethasone that can be injected into the intravitreal space, and eventually they will keep degrading, but also releasing dexamethasone over time locally. Now, a corticosteroid like dexamethasone has a lot of side effects when taken orally. It's not a good idea to take it for a very long period of time, but when you are directly putting it in the site of action and when it is released over controlled time in a controlled manner over a specific time period, then you sort of reduce the side effects associated with some of these drugs. So, that is a good segue now for us to come to the last part of the lecture, where we are going to look at one quick study where the use of several of these polymers that we have just looked at, namely polycaprolactone, polylactide, co-glycolide and PLLA for the controlled release of dexamethasone. This is published work from our lab, but what we will do here is we will quickly look at different factors that govern drug release mechanisms based on which property guides drug release. So, what was done here was dexamethasone was taken and put into a series of different types of electrospun materials. So, these polymers were prepared in the form of nanofibers through this process called electrospinning, which is a polymer processing technique which we will have a chance to look at in more detail in a later lecture. But essentially at the end of it, you get these drug impregnated nanofibers, if you will. And here our interest was to look at what types of factors govern the release of this one drug dexamethasone from these different matrices. So, all the release curves you see are all from polymers that are polyesters, polycaprolactone, PLGA and PLLA, and yet you see you get a very wide spectrum of release profiles. So, one can go back and ask what factors contribute to it, and we will do a quick snapshot of some of these factors. So, first looking at bulk physical properties, we see that these two first, in fact all these three are PLGAs. So, what we did here is this PLLA and this PCl, which you are familiar with, 85-15 and 50-50 refer to the PLGA ratios, the lactide to glycolide ratios in PLGA. So, these two are PLGA 50-50, this is PLGA 85-15. Now, 85-15 is going to degrade slower, right away you can sort of sense that, and 50-50 is going to degrade faster, and you can quickly see how that will have an effect on release profiles. In addition, what we also did is change the fiber diameter of one of these polymers which is 50-50. So, you see that the average fiber diameter of the HT high diameter 50-50 is significantly higher compared to all the others which are all similar in diameter. So, you see first that the porosities are all comparable. Electrosponge meshes are largely consisting of air. They are very porous, and by characterizing through electron micrographs, we characterized the diameter, and we showed that the HT version, high diameter version, was indeed much higher in diameter compared to the low diameter versions. We also determined based on swelling in different solvents a way to quantify bulk hydrophobicity, because the drug release is going to happen from these fibers. We also wanted to quantify differences in bulk hydrophobicity. So, you see right away based on the fiber diameter differences, the black and the red curves are essentially the same chemistry. They are both PLGA 50-50. One is a lower diameter polymer, the other is a higher diameter polymer. Now, you see that the LD, the lower diameter version, releases the drug faster initially compared to the high diameter fiber, and then eventually they catch up towards the end of the study. Now, these initial differences may be attributed both to the difference in the diffusional path length of the drug for the low diameter fiber versus the high diameter fiber, as well as differences in the drug polymer ratio. If the drug loading, the amount of drug that is put in is kept constant, and in one case you increase the amount of polymer, the drug polymer ratio also changes. So, that explains why you might get these differences in the initial time periods. But you also see that the 8515, which does not degrade as fast, is releasing the drug at near constant rate. This is what you would typically call a near zero order kind of a release profile. And polycaprolactone and PLLA, which are both semicrystalline and essentially non-degradable as far as the time period of the study is concerned, one releases very rapidly, one releases almost nothing, essentially very slow release from PLLA. So, then you can go and ask, why do we see these differences? So, for the amorphous degrading polymers, what you can sort of see is, again, this is just a snapshot of the study itself. What you can see is, you can track the degradation over time, and you see that both PLLA and PCl, as expected, which are semicrystalline, do not show any degradation with time over 28 days. But the 50-50 essentially by the end of the study becomes fragmented, and shown here are just micrographs from after 7 days of incubation in just phosphate buffered saline. So, we see differences not just in degradation rates here, we also have differences in matrix architecture initially, which is the fiber diameter. Then we have fiber swelling occurring due to uptake of PBS and mesh degradation in the later stages, all of which can help to a reasonable extent explain the release from the PLGAs. Of course, PLGA is also unique in that it has a glass transition temperature slightly above 37 degrees Celsius, which means at a body temperature of 37, the PLGA chains are glass-like, which means the free volume available for the drug to diffuse out is going to be quite low. So, that also can help one reason why you see near constant release, because of PLGA's glass transition, its glassy nature, and availability of free volume and the diffusivity of the drug. So, that is the connection you have to make. But this still does not fully explain why PCl, PLLA, which do not degrade much, release, one of them releases the drug very rapidly, one of them does not show any appreciable release over the time period that we have studied. Of course, we also saw that the PCl is the most hydrophobic in this experiment based on quantification of relative hydrophobicity using differential swelling of the polymers in different solvents with different polarities. And one would expect that because the drug is also hydrophobic, some of these interactions may be stabilized, and yet you see that PCl is the one that releases the drug the quickest. So, probe a little bit deeper, and what you will see is that if you investigate the properties like Tg, Tm and delta Hm, which is the glass transition temperature, the melting temperature and the enthalpy of melting, we see that in the case of the PCl, there are no appreciable shifts in the glass transition or the melting temperature, whereas in the case of PLLA, we see shifts between the polymer that is not loaded and the polymer that is loaded with the drug. So, those in turn indicate the presence of possible interactions stabilizing the drug in the amorphous state in PLLA, which led to very slow release, also suggesting the potential absence of interactions in the case of polycaprolactone. These are also supported by investigation of Hansen solubility parameters, where we are showing here the delta, the difference in the Hansen solubility parameters for the DEX and the different polymers. When you take PCl and DEX, you see that the number is 9.4, but for all the others, PLGA 50/50, 85/15 and PLLA, the numbers are much lower, and typically numbers below 7 or so are considered miscible, and between 7 and 10 are typically considered partially miscible. So, if you have the closer the Hansen solubility parameters are, the values are for the drug and the polymer, the more miscible they are. If you remember, like dissolves like is what we have seen in lecture 14 when we did Hildebrand and Hansen. So, here is a direct application of that in controlled release. And the farther apart they are, the less likely they are to be miscible. And that sort of may help explain why the PCl, the drug is not that stable in PCl, and which is also supported by the presence of these drug crystallites on the surface of PCl as opposed to a clean surface that we note for PLLA. So, essentially what this tells you is that in one case, namely the PCl, because of incompatibilities, the drug could have partially been miscible and could have phase separated onto the surface, resulting in very rapid release, whereas with much better Hansen solubility parameters also supported by changes in Tg and Tm, which indicate drug polymer interactions, the drug could have released much slower from PLLA. So, in this one study, we have revisited our concepts for microstructure, namely crystallinity and amorphous. We have looked at effects of degradation, we have looked at effects of Tg and Tm, we have looked at the effects of Hansen solubility parameters, all in the context of controlled drug release. So, with that, I hope you get a really good sense of how synthetic biomedical polymers in those kinds of materials, the structure, property, function, paradigm directly influences how these polymers are employed in specific biomedical or clinical applications.